Good afternoon, everyone. It's just about 1 o'clock, so I'm going to start my introduction here. Welcome to the Invasive Species Center's webinar series. My name is Mccannii and I'm a Program Development Coordinator at the Invasive Species Center, and I'm going to be your moderator for today. Before we get started, I would like to acknowledge that I'm speaking to you today from the Robinson-Huron Treaty Territory, the traditional territory of the Anishinaabe, home of Garden River First Nation, Batchewana First Nation, and the Métis Nation. The Invasive Species Center is a not-for-profit organization that works to prevent the spread of invasive species in Canada and beyond. Again, before we get started, I just have a couple of housekeeping items that I'd like to mention. There will be time for questions at the end of the webinar, so at any time throughout the webinar, if you think of any questions, you can go ahead and type it into the question box, and I'll be reading the questions to the presenter after the webinar. If you're having technical difficulties at any time, you can type them into the chat box or send me an email, which I have left at the bottom of this slide for you, and we can try to resolve your issues for you. Lastly, there will be a brief survey following the webinar, and if you could take a few moments to fill that out, we'd greatly appreciate it. Now, I'm very excited to introduce today's presenter, Dr. Sharon Reed. Sharon, thank you for taking the time with us today to share some of your work on beech leaf disease. Dr. Sharon Reed has been working as the Forest Health Research Scientist for the Ministry of Natural Resources and Forestry for nearly 3 years. The lab's current efforts are focused on detection, survey, and management of invasive species. Their topics include oak wilt, beech leaf disease, and Phytophthora. So with that, I'm going to hand over control to Sharon, and she can take it away from here.
Hello, everyone. It's a real pleasure to be able to speak to you today about beech leaf disease. Oh, it's very sensitive. Let's see.
Do you still have control, Sharon?
I do not.
Should be good to go there.
Okay, now it is moving again. Okay, let's see if I can. All right. So first I wanted to go over the symptoms associated with beech leaf disease, just in case some of you are looking for it in the field. And so the characteristic symptom is the photo on my right-hand side that has the 2 arrows. And you notice that there's dark spaces in between the leaf veins. So in between the leaf veins, bands develop. It doesn't have to fill up that entire space between those smaller leaf veins. Sometimes the band will get thinner as it goes out towards the edge of the leaf, or it only fills up half of the leaf band. Hopefully you see my pointer here. So it doesn't have, the stripe does not have to go, the entire extent of the area. And it's easiest to see this diagnostic feature when the light is shining through the leaves. So essentially, if you're looking up at the leaf and the light is coming through it, that's when it's easiest to see this banding pattern that occurs. If you look at the top of the leaves, you're going to see something more like this other picture where the leaves are have a more puckered look to them, with the bands raised slightly in between the leaf veins. All right, some other photos of symptoms. So the very first photo, these are normal leaves that have just emerged from the buds in the springtime. To the next to that, These are leaves with the infection. And here the bands, instead of being that dark green color, have a reddish tone to them. And that will darken as that leaf matures. The symptoms, when a tree has been infected for multiple years, we start to see what we call more severe symptoms. In that case, those dark striped bands will become chlorotic. They'll have a yellowish color to them. The leaf will become very thick, have a coarse, almost leather-like constitution to it. You can start getting tears in the leaf tissue and some curling as well. Sometimes the leaves will have these, it's almost blister-like when you're looking at the top of the leaves as well. It's just kind of an raised up area in between the leaf veins. Oh, just try to get back. I just got these photos last night. Thank you to Rebecca Lidster for these photos. So I wanted to show you what these symptoms look like in the fall at this time of year. So you can still see these bands. The rest of the leaf has developed its usual fall color, but these bands are either very dark or kind of a more the summer dark green look to them. Here, this leaf here that has a very, very light infection, that band's very light in coloration, but still that more of a summer green than the fall coloration that's on the rest of the leaves. Another thing that occurs with beech leaf disease is that there is some bud abortion and early leaf shed. So the branches are more bare than you would normally expect on in comparison to the healthy branches. All right. It's not just the saplings that are affected by beech leaf disease. The disease does affect the trees in the overstory. This is a photo taken from the US in the area where beech leaf disease was first reported. We also have some locations in southern Ontario that are looking similar to this at this time. The symptoms of beech leaf disease are visible in the winter as well. Beech is one of those trees that holds on to some of its leaves during the winter time. So you can see the stripes in between those leaf veins again here and here. You can see it on the leaves on the ground as well, although it gets harder to determine if the symptoms are due to beech leaf disease or some other infection or insect damage. Okay. There are other insects, other damage on these beech leaves that can be confused with beech leaf disease. The Iranian patch developed from mites, can be many different colors, brown, orange, red. It's often along either the midrib or along these small veins. So that can be confusing, but it's more raised up than the beech leaf disease is. So with some experience and looking at photos, you should be able to determine the difference between those. There's also, there's also powdery mildew is sometimes confused with beech leaf disease, just because powdery mildew can cause that yellow chlorotic coloration. But powdery mildew occurs in a circular pattern, and it crosses over the leaf veins. Here's a good example of kind of a circle, and it goes over a number of leaf veins, not in between the leaf veins like beech leaf disease. And woolly beech aphid damage might be one of the closest lookalikes, in my opinion. It does have a slightly more stipuled look to it, whereas beech leaf disease, that discoloration is more of a continuous band between the leaf veins. We are asking for samples to be sent into the lab for diagnostic purposes because there's often multiple types of damage on these leaves. For example, this leaf in this photo has both anthracnose and beech leaf disease. That can cause confusion about if beech leaf disease is really present. Great. So this slide comes from the Cleveland Metro Parks, and they followed the symptom progression over time for one season for this study, and they found that the damage from beech leaf disease occurs in the buds. Once a leaf unfurls, the damage that's on that leaf is the damage that it will have for the rest of the year. The things that do change during the year is that the branches that are more severely affected by beech leaf disease, those leaves can have early leaf drop as early as June. And the necrosis, or the dead material that develops on the outside of the edge of the leaf for the leaves that are severely impacted, that can develop over the summer as well. But for the most part, the symptoms that you see on a leaf in the spring are the same symptoms that you would see on that same leaf in the fall. Okay. Okay. All right, so this is another slide from the Cleveland Metro Parks, and they have a series of plots that are set up to study vegetation changes. And it just happened that those plots became infected with beech leaf disease. So they've been able to monitor the changes in the leaf cover over time for a 5-year period. And they've been able to document the change in the amount of normal leaf coverage. So at the beginning of their study, they had 149 individuals here. The majority of the leaves were normal. The following year, they're dropping down to about 85% of the leaves being normal. Then by year 3, 60%. Year 4, 40%, year 5, they're down to 20% of the leaves appearing normal. So there's a quick change in the cover and the disease progression over a number of years. All right. So beech leaf disease was first detected in Lake County, Ohio, just south of Ontario. This little yellow patch here on the map. And then as they started surveying it, they found it was not only in Ohio, but in Pennsylvania and New York. And about the same time in 2017, we became aware of beech leaf disease through a flyer from the Ohio Department of Natural Resources. And we had some samples that had been sent in that were suspect and we determined that it indeed was the same symptoms as in that flyer, and that it was beech leaf disease. And that was in Elgin in Norfolk County. Since then, we've been able to detect it throughout most of the ecoregion 7E, or the Carolinian forest. Last year, we had an added detection in Lambton County, in Toronto, and the Niagara area. And this year, with COVID, we weren't able to do a lot of survey for beech leaf disease, but I was able to find a report, a photo on iNaturalist of a beech tree, and I noticed that it had beech leaf disease symptoms. And Essex Conservation Authority was willing to send in a sample to us, so we're able to confirm also that beech leaf disease is present in Essex County as well. There have been several new detections of beech leaf disease this year in the states, including Rhode Island, Massachusetts, and New Jersey. I know that there will be more reports made soon. The deadline this year for reports to go in to the metro parks who are doing the map, keeping track of all the mapping data, is November 1st. If you're interested in an updated map, I would check the don'tmovefirewood.org map for an updated map during the winter to see all the locations that have beech leaf disease after this past survey year. So beech leaf disease does kill trees. Mortality of understory trees has been reported. It's in the same area as where the disease started, so that disease has now been documented in this area for about 7 to 8 years, and that's where they're reporting the mortality. On this map, the darker the green color, the more severe the symptoms. So you can see that general pattern that the darker colors are here. The situation is more severe here and less severe along the outside edges. Oops. Okay. All right. So beech leaf disease is known to affect multiple species of beech trees. We have reported it here in Ontario on American beech as well as European beech, but it has also been found in some arboretums in Ohio, and they reported that it also infects Oriental beech and Chinese beech as well. So there could be some regulatory issues in the future because European beech does cover a wide area in Europe, as well as the Oriental beech, as shown in the map here. The green is the European beech, this peach color is Oriental beech, and the triangles are the locations where the species are introduced and naturalized. All right. So the causal agent for beech leaf disease, we've learned a lot since 2012 when it was initially found, especially since 2018 when when a research group was formed to look at beech leaf disease specifically. Initially it was thought that it must be some type of fungus, a bacteria, virus, or a phytoplasma. So a lot of individuals have spent quite a bit of time looking at these different aspects, and no consistent patterns were found. Then in 2018, Dave McCann from the Ohio Department of Agriculture reported that he was seeing a nematode associated with the diseased leaves. And at that same time, there were reports that a nematode was causing similar symptoms on Japanese beech, and that newly described nematode was named Litylenchus crenatae. So after doing some morphological work, this new nematode that has been found to be associated with the American beech in the US and beech leaf disease has been named Litylenchus crenatae subspecies Mccannii, after Dave McCann. You can see the symptoms, that darkening or change in color between the leaf bands is present on these Japanese beech that are infected with Litylenchus crenatae. You can see that puckering as well here. So similar, but some differences as well, not identical symptoms. And the damage has been associated with other nematodes that are closely related to Litylenchus crenatae. So there's this Dilenchus leptosoma, Zeadolenchus pedosporum, and Litylenchus comprosma. Right. But this group of nematodes, there's not a lot known about them for the Litylenchus. Now there are just 3 species in this genus. So after learning about the nematode and finding it associated with these leaves here in Ontario and also in the US, we started to do some pathogen trials to see if the nematode, exposing the leaves to the nematode would cause the symptoms. And we were able to find that if the leaves and buds were sprayed in the fall, and really the important part is spraying the buds with a solution containing the nematode, then in the springtime, those leaves would leaf out with the beech leaf disease symptoms. The buds that were sprayed with just the water solution did not develop the symptoms. And in Ohio, they've continued on with this work to learn more about this infection process. Right, here's some photos. These photos are from Lynn Carta, works for the USDA ARS, and also David Burke from Holden Arboretum. So photos of the Litylenchus nematode here, and the pink staining of the nematode in the tissues of the leaf material on this slide. This is a photo of the Litylenchus nematodes coiled together inside the bud. And here's the bud that's been cut open for the imaging. And then also here's a photo of a petri dish with the that's filled with water and has strips of the beech leaves, infected beech leaves in it. And so the nematodes actually just leave. Once you cut up the leaf and put it in the water, the adult nematodes will just leave that leaf tissue and move out into the water. So that's how we count how many nematodes are present. So when we learned about the nematode in 2018, we asked for samples to be sent up to us. And we did find the nematode shortly after that. It was just surprising. It was kind of like, it was so obvious. Why didn't we see this before? But we had been looking at fungi and some other items. It's very uncommon for a nematode to be associated with a disease of trees. So we wanted to see if the nematode was associated with symptomatic trees at all the locations, and if it was associated with symptomatic leaves, if it was found in asymptomatic leaves. And we wanted to know more about the time of year and the types of tissues that we should be sampling, both for detection purposes and also for being able to carry out research projects. So we set up research sites with the help of Forest Health Research Monitoring, or Forest Health Monitoring Group, and looked at 3 locations that had beech leaf disease and 3 locations did not have beech leaf disease. And we also looked at leaves on trees that were symptomatic and leaves on trees that are asymptomatic. And we checked the leaves every once a month, essentially, following it through time for 18 months. Okay, so just focus on the black bars, which are the symptomatic leaf samples from June to October. So initially, we only found small numbers of the nematodes in the leaf material, and then the numbers started to increase over time. So the nematodes are able to complete the reproductive cycle in a month. So that helps explain why we're seeing such large numbers of nematodes in the fall. And in the beginning of the summer in June, and it's not necessarily that there aren't that many nematodes present in the leaves. We only looked at adults, so there are likely a lot of nematodes in the egg phase present as well. So these numbers are probably an underestimate of how many nematodes are in that leaf material. Switching over to the asymptomatic leaves. So these are asymptomatic leaves that are on a symptomatic tree. Okay. So in June and July and August, we didn't find any nematodes in the leaf material. And then all of a sudden, in September and October, we were finding very small numbers of nematodes. So this could suggest that there's some migration of the nematodes from the symptomatic material to the asymptomatic material sometime in August. Right now, we don't know that much about how this nematode is moving around. Some foliar nematodes are able to move in films of water from one place to the others. Others are moved around by mites and other animals. So they are able to move, but we're not sure how it's moving in this particular situation. In the winter, we did something similar, but a slight change. We looked at buds versus ground leaves versus attached leaves in the winter. If you look at the buds, we found nematodes in the buds all winter long. So this means that there are live adults that are capable of emerging from leaf tissue in the lab that are present even though it's cold in the winter time. We also found the nematode in the ground leaves. In fact, our largest numbers were in November in the fall from those ground leaves, but then the numbers reduce after that once we start getting snowfall and it's more cold, but still we were consistently able to find nematodes throughout the winter and spring months. The attached leaf material, that was the one type of material where the numbers of nematodes decreased greatly over time, and then by the end of the winter, we were not finding nematodes in the attached leaves. And I would also say that we were running out of attached leaves. It does seem like the diseased trees don't keep their leaves through the winter as much. Okay, and we are also able to answer our questions about, if you want to take a sample and confirm the presence of the nematode, or you're doing research and you want to collect collect samples and show the nematodes present, when should you collect the samples? So for the symptomatic leaves, we found that in the first year, June through October, we were getting good numbers of nematodes or good detection rates all through the summer, even when the nematode numbers were smaller. The one time of year that the detection rate was particularly low was in May of 2019. And I would say that, if you're collecting a sample for beech leaf disease, you want to send it into a lab for confirmation, or you're going to look for the nematode for research, then I would not collect those samples in May or early June, because that's usually when we have to ask for a second sample later in the season to make the confirmation of the nematode. For the winter time, we were able to find, we were able to detect the nematode reliably all winter, as long as we were looking at the buds and the detached leaves that were on the ground, but I would avoid sampling from attached leaves. I also wanted to mention that during this study, we did find the nematode at several sites that had either very little beech leaf disease or no detectable beech leaf disease. And those detections were made from ground leaves or leaves on the ground during the winter time. So I have some thought that it might be a better time of year to look for the nematode if you're trying to confirm the presence of the nematode at a place that does not appear to have symptoms in the wintertime when all the leaves are on the ground, so that we can sample from the entire overstory. All right. So last winter, seems like it was such a long time ago, before COVID, we were able to look at this question about where did this nematode come from. We know from working with Lynn Carta that this type of nematode has only been reported from Japan, Korea, and New Zealand. So the thought is that this species could be from the Pacific Rim area. We are wondering, has this nematode been introduced from many different locations, or is it a single introduction, or maybe multiple introductions but from one area? So we were able to work with Katrin Fitza, who was a visiting scientist, a postdoc, visiting Jeremy Allison at the Natural Resources Canada, and to work with her advisor, Bernard Slippers, from the University of Pretoria, to better answer that question. And we had collected leaf samples from locations throughout southern Ontario and collected the nematode from those leaves. And we looked at the DNA of the nematodes in 3 different gene regions, and we found that all of these nematodes had identical sequences, which would suggest that they are from have come from a single population. So it could be that the nematode has been introduced multiple times from one region, or a single time from one region, but it's probably not been introduced from many different locations, many different times. Right. Last year, we were also able to set up beech leaf disease impact plots. And although that we're mostly interested in the impact of beech leaf disease, we also set up a study to look at scale and beech bark disease, because these are really important pests of beech throughout Ontario. So these plots that were set up last year will be able to monitor symptom progression over time, dieback and mortality, as well as regeneration of beech and regeneration of other tree species. This is an international project. These plots are being set up throughout the eastern United States in places that do and don't have beech leaf disease. And one of our interests is that this could be a first look at not only how beech leaf disease will impact forests, the Carolinian forest, but all the Great Lakes, St. Lawrence forests, in some of our industrial forests that have been heavily impacted by beech bark disease. All right, so I'm going to take a step back from beech leaf disease and just talk about beech bark disease for a few minutes because it's such an important pest of the beech trees. So in the Great Lakes St. Lawrence, specifically on the Canadian Shield, beech bark disease is present, and it's in the killing front and advancing front stage with some development of aftermath forest. So beech bark disease occurs when an invasive scale attacks and feeds on these trees, and that damage from the scales can then be colonized by a native neonectaria fungus that kills the bark on the tree, and eventually, if the cankers are large enough, then it shuts off the flow of nutrients in the tree and can kill it. And the mortality rate from beech bark disease is quite high. Estimates are that 99% of beech trees die from beech bark disease eventually. And one of the problems with beech bark disease has been that when the overstory beech die, then they send up a lot of sprouts from the root system, and these sprouts are also susceptible to the disease. So as they grow, they become infected. Meanwhile, they are shading out all the other tree species that are on the forest floor. So you get these diseased thickets of beech trees in the aftermath forest, which can be an issue, especially management-wise. In the Carolinian forest, we have not seen that same development, or at least it's not occurring at the same rate. Scale was first reported in 1966 in Elgin County, and beech bark disease was reported as early as the 1980s in Toronto. But today, there are still just pockets of beech bark disease throughout this forest that's currently affected by beech leaf disease. And it's a much slower progression. So we're interested in knowing, we know what happens when you just have beech bark disease. So what happens when you have beech leaf disease and beech bark disease co-occurring and developing at the same time? So we were able to set up plots at 17 locations in Southern Ontario last year. It turned out that both beech bark disease and beech leaf disease were spread farther than we thought when we went down to set up the plots. We ended up with 4 locations that had low severity symptoms of beech leaf disease, 4 locations that had higher severity symptoms of beech leaf disease, 5 locations with both beech leaf disease and beech bark disease, and 4 locations with just beech bark disease. And each one of these locations has 2 plots. So this slide, the numbers are the percentage of trees that were infected with beech leaf disease. So in the stars are the 2 locations where beech leaf disease was first reported in Elgin County and Norfolk County. So you can see that the numbers, like here's 100% of the trees in those plots were infected, 98%, 94% of the trees infected. So around the area where we first noticed beech leaf disease, we had the highest percentages of trees infected. As you move to the north, west, and east, we had far fewer trees infected with beech leaf disease. Okay, this next map is the severity of the beech leaf disease infection. So severity ranged from 73% of the canopy affected by beech leaf disease down to 0% of the canopy affected by beech leaf disease. And again, that severity was higher in that initial area where we first detected beech leaf disease. Okay. So we found that multiple pests are affecting beech trees of all sizes. This first row is the percentage of trees with beech leaf disease. So you can see that it's affecting the saplings in the understory, about 40%. Slightly more effect on the intermediate suppressed trees. So these are the trees that are the lower portion of the overstory, and still more than 40% of the largest trees in the forest being affected by beech leaf disease. The scale was much more widespread in the study, with the overstory being more infested with scale than the saplings. Upwards of 80% of the trees in the study, and there were 666 beech trees in the study, compared to slightly more than 40% of the beech trees in this understory affected by beech leaf disease. And then this last row here is trees infected with Neonectaria. So essentially, these are the trees that had beech bark disease. And there were 14 trees in the study that had beech bark disease, and most of those were in the upper overstory, which is common for beech bark disease. The reason why I wanted to put this slide in here was to talk about the fact that these trees are being attacked by multiple pests. And so the overstory trees, beech leaf disease has not been associated with mortality of overstory trees in the Ohio area where it's been, most of the study has been done. But here in Ontario, the situation is a little bit different, where there's a lot more scale present. So we'll see what happens with these overstory trees that are being impacted both by the scale and beech leaf disease. And unfortunately, beech leaf disease does affect the seedlings as well. We found beech leaf disease on the seedlings in 18% of the plots. So that's about 34% of the 208 seedlings in the study. So this brings about the question of what does the future hold for beech, and not only beech, but for the forests that contain beech. So there are some assumptions that we will see mortality of saplings and seedlings similar to what's been seen in Ohio over the course of the next several years, in areas that have had at least 5 to 6 years of symptoms. Also assuming that more light will be reaching the understory because of that early leaf drop that occurs on those severely affected trees. Also, the severely affected leaves do tend to be smaller, which can allow more light through the canopy. And then the assumption that the increased number of pests will result in more mortality of beech overstory trees. That's something hopefully that this study will be able to reveal for us, how much mortality will occur, and if the stress of the scale and beech leaf disease could increase the amount of beech bark disease in the forest as well. But I can, even though I can't tell you the exact future, I can tell you what is in the sapling layer and the seedling layer that will probably respond to the increased light that we are expecting. So the saplings, we found that other than American beech, there was a lot of sugar maple and white ash. And of course, sugar maple and white ash were not just abundant, but they were very common among the different locations. And of these two, most likely it'll be the sugar maple that perseveres. since we have EAB2 that will attack white ash in the future. In the seedling layer, it's the same species, but just a different order of importance. So sugar maple was the most frequently occurring species in the seedling layer, and the most abundant, followed by white ash and then American beech. Right, so we would suggest that there will be more maple likely replacing the beech overstory if we do start to lose those trees. Okay, I wanted to take the last few minutes just to give you some updates from other researchers so that you know some of the other work projects that are being carried out. All right, so last year, beech leaf disease was found in Connecticut. So Bob Mara from the Connecticut Agricultural Experimental Station has joined in the research group and has been setting up long-term monitoring plots. Is also working to develop forensic molecular markers to look at the pathways of dispersal and also likely sources of new infections. Oh. And he sent me a map of all his locations. He's doing quite a bit of work setting up plots. So all of these red little pins that are dropped, those are beech leaf disease positive locations in Connecticut. So you can see it's spread all along the Atlantic front. And the blue are their long-term monitoring plots. And I'm excited because they have a lot of these long-term monitoring plots established. So I think it'll be great to see the results of their data and be able to compare that to what is happening in Ontario in the future. Dr. Lynn Carta from the USDA ARS and Ron Okwa are looking more closely at the potential for mites to be transporting the nematode. So they have reported finding the Litylenchus nematode on mites, and I think that they should have a research paper out soon discussing that. And they're also doing some studies with predatory mites as well. This research just actually came out in 2020, performed by David Burke, Adam Hoke, and Jennifer Koch. The work was done at Holden Arboretum in Ohio, and they are asking questions about, so we know the nematode is associated with beech leaf disease. Are there any microbes, bacterias, or viruses that are fungi? Actually, they look specifically at bacteria or fungi. Are there any associated with nematodes? Because nematodes often will have microbes associated with them that cause disease, or the damage that they cause results in allowing other microbes to cause damage. So they did find a difference between symptomatic and asymptomatic leaf communities. In the bacterial communities were slightly different. So they're looking a little bit more carefully at this Wolbachia, which can be associated with arthropods. And they also found this bacteria called Mucilaginibacter, which is able to degrade pectin and polysaccharides. So those, the thought in their paper was that this Mucilaginibacter could be contributing to the symptoms that we see associated with beech leaf disease. All right, there's a team at Ohio State. Carrie Ewing is the doctoral student doing the research. This team is also looking at all the DNA associated with symptomatic materials and asymptomatic materials, and determining if there are any differences in the DNA for fungi, bacteria, phytoplasmas, viruses, and nematodes. and they are interested. Some of their results, they also found this Wolbachia bacteria to be different between the symptomatic and asymptomatic leaves. But they have a list of a few other microbes, Pseudomonas, Erwinia, Xanthomonas, that they think could be associated with the symptoms. And I know that they had planned to do some disease trials this summer, and I think everything's on delay due to COVID. So hopefully they'll be able to carry out this research in the future. And there are ongoing treatment trials. So these treatments would just be for high value beech trees. This work is being done at Cleveland Metro Parks and Holden Arboretum. It's being done with industry involvement. They know that they've been doing some injections, and some of the products that are being looked at, for example, emamectin benzoate, which is a nematicide, and potassium phosphate, which is thought to improve tree health. These are chemicals that are being trialed to see if they either reduce the nematode or reduce symptoms and increase tree vigor. Unfortunately, at this point, it's too early to tell if any of these treatments will be effective, but hopefully we'll find out some positive results in the future. All right. So that is my talk. I do want to say thank you to the forest health monitoring technicians from the Ministry of Natural Resources and Forestry. They've put in a lot of time collecting samples and sending them to our lab, as well as Sylvia Greifenhagen for counting all the nematodes. Dr. Yu from Agriculture and Agri-Food Canada, he did a lot of early work on the morphology of the nematode, doing taxonomic identifications and helping us understand the best way to extract the nematodes from the leaf material. Also others such as David Dukatowicz, Jeremy Allison, the many conservation authorities, the Canadian Nature Conservancy, Ontario Nature, Ontario Parks, and the Thames Talmot Land Trust for allowing us to put plots on their properties. And then all the other researchers who are part of the larger international research group. All right, so that is my talk. Thank you very much.
That's great, Sharon. Thank you so much for that wonderful and informative talk. I'm going to open up the question box here, see if we have some questions for you. Okay. So Simon asked, what is the vector for this nematode, if you know?
So that is something that's being researched. So there's evidence that there could be mite transfer. There's also been suggested that because this disease appears to be moving so rapidly that there could be bird involvement as well. Those are two areas that are being investigated.
That's great, thanks, Sharon. And I have one more question here from Dimitri. Oh, a couple more are coming in, so I'll just read those. Considering beech regeneration is now the predominant species in the understories of states and provinces well into the aftermath of beech bark disease, do you think that beech leaf disease will assist in eliminating beech thickets?
So if beech leaf disease is able to spread that far north, I do think that there's a possibility that it will limit the thickets, just because we've seen that symptom severity in the development of the symptoms, how quickly they develop seems to be worse in the understory.
Okay, thanks, Sharon. So John is asking, was beech leaf disease found exclusively in natural and rural beech stands, or were there detections in urban environments as well?
So in Ontario, our detections do tend to be in the natural areas. The detection in Toronto was actually a European beech hedge that was infected with beech leaf disease. It is being seen in the urban areas, but it just hasn't been reported as much from those locations.
Okay, thank you. Sharon is asking where samples can be sent for analysis.
So our lab is the only lab I know of that is currently accepting beech leaf disease samples, and we are interested in receiving samples from counties where beech leaf disease has not been detected yet. So you could contact me or a member of the monitoring team to find out more about sending in a sample.
Okay, that's great. Great information for people to have. Okay, there's a few more questions, Sharon. I hope you're up for it. Do you have any recommendations for practitioners to avoid spreading the disease to other sites via equipment, clothing, etc., or other management recommendations?
That's a good question. In general, I think it's definitely not to move leaves around because we have been able to confirm that they survive, the nematode survives in those beech leaves on the forest floor. The difficulty is that we don't know how the nematode is being moved right now, if it's through arthropods or through birds. So I guess my general recommendation would just to follow basic practices of keeping your shoes clean and not moving material from an area where there's an infection to an area where there's not.
Great, I think that's great advice, Sharon. And I think that may have answered the next question as well. William asked if there were any precautions that need to be taken while traveling between survey plots. So I think the same recommendations would apply to that.
Yes.
Simon asks how you inoculated the beech seedlings under greenhouse conditions. Did you inject nematode populations into the leaves?
So no, actually, it was a greenhouse study, and we actually sprayed the nematode using a water solution onto the leaves and the buds.
Okay, just have a couple more questions. Have you seen any infected trees along provincial corridors?
So, I mostly rely on the monitoring team, so I don't feel that I can answer that question very well. I don't think there's any evidence that beech leaf disease is being moved along corridors specifically.
Okay, thank you. If we find leaves that look suspicious for beech leaf disease, how should we preserve them for shipping? Is it okay to just ship dry in an envelope?
Okay, so how we usually have them shipped to us is we ask that you collect enough leaves to fill a sandwich bag, a Ziploc bag, so to have the bag ziplocked, and then placed in a small cooler with some ice packs, and then the cooler needs to be taped shut, so that way if there was anything in there, it stays sealed inside of that container until we get it to our containment area of our lab.
Okay, great. And that was the last question, Sharon. So thank you for answering, and thank you to everyone for those great questions. So just to close, I would like to thank Dr. Sharon Reed again for presenting today, and to thank all of you for tuning in. This webinar will be recorded and it will be posted on our website, www.invasivespeciescenter.ca. And just a reminder to please take a couple of minutes to fill out our survey. We'd really appreciate that. And our next webinar will be on Wednesday, November 25th. And that will be covering new biological control agents for the management of invasive plants in Canada. So hope to see you then. Thank you again for tuning in today, and thank you, Sharon, for your time. I hope everyone has a wonderful afternoon.